So, hello everyone. It's a pleasure to be here, and I would like to thank the organizer for this event for inviting me to present an overview of our research activity in the EPION team, which is a team dedicated to the development of methods for modeling of biomedical information, in particular with a focus on medical imaging data. And so among the different applications we focus on, we have the problem of modeling the evolution of neurodegenerative disorders. So neurodegenerative disorders are pathologies that develop over the course of several years, decades, and affect the human body at different levels, notably in the brain, so they manifest through brain atrophy, but not only. We have a functional impairment, changes, biomechanical changes, so the molecular level, and all of these so describes different trajectories of evolution of different markers over time, which also involve other kind of biomedical information, which could be clinical scores, there are genetics determinants of the disease, and then researchers are currently interested in studying vascularity and up to microbiome effects on the evolution of this pathology. So what we would like to do is to able to develop methods, since we currently don't have a definitive cure against the disease. So we are talking about a disorder that evolves over the time, it cannot revert. And so first we would like to better understand the dynamics of these pathologies, so how all these ingredients, how all these factors interact together to describe evolution of the pathology, to identify better markers of the disease progression, and better understand the efficacy of drugs. And all of this is to develop a new hypothesis on how and when we should give a drug, and imagine an intervention to maximize the benefits that the patient could have to modify these pathological trajectories. And so I will give you just an overview during this talk on some of the aspects we currently focus on, which is basically how to model disease progression, integrating different kind of data, but also different, increasingly complex hypotheses on the dynamics of evolution and co-evolution of different biomedical information. So the basics is what we call probabilistic disease progression modeling. So basically we have hypothetical models of the evolution of the disorder. So we have ideally these different aspects that evolve over time with different trajectories, different dynamics. But in reality, what we see are patients followed over short time spans, so a couple of years, with very noisy measurements and a large heterogeneity of observation across patient, across biomarker. So what we would like to do is to go from the real measurements, so what we see on the right, being able to reconstruct long-term evolution that spans lifetime. And the problem is that we don't have currently cohorts, large cohorts, to model the evolution of the disease from the early stages, like people in their 50s, 60s, up to actually the latest stages of the disease. So how to do? Well, from a modeling perspective, you can imagine that we have different measures of patients over time, like for example, during a clinical trial. And so we can make, to reconstruct a long-term pathological evolution, we can make some hypotheses, in particular that the disease is a smooth process over time, so we don't have sudden changes, for example, of the brain shape during the pathology, and also is a monotonic process, so there is no way back once the disease starts developing. And so based on that, for example, we can try to identify parameters to reparameterize the trajectories, so observation for different patients, in order to identify the best curve that satisfies our constraint. For example, that is smooth and monotonic, that can describe optimally this different observation. And then we can interpret the parameters as from a clinical point of view, for example, as a disease stage. And so this kind of approach defines what we call a disease progression model. And here I can see the disease progression model in action. This is just a dummy illustration. So we have different biomarkers, which we start from short-term data, which are very noisy and define a short-term time axis. And what the model is trying to do is to refine x-axis and moving the observation for all the biomarkers for a given subject across this hypothetical new estimated time axis to identify monotonic trajectories over a long-term progression progression scales. And so in this way, we can basically identify, if we apply this on real-world data, we can identify different trajectories associated. For example, here is a cohort of individuals that represent different stages of the pathology, from healthy, mild cognitive impairment, up to Alzheimer's disease. And we see that even though the original data was observed over 5 years, the model is able to recover trajectories of different kinds of biomarkers over 10 years, or 20 years, sorry, and showing also the differential relation of the evolution of different biomarkers, showing, for example, that we have brain changes that happen first with a steeper slope, for example, for the temporal region in the brain, while cognition starts slowly and then accelerates toward the later stages of the disease, which is clinical plausible. And then we can align, so project the subject along this new timeline based on their observation, and we can see that we can discriminate quite well groups across the continuum of the disease. So we can use this model to integrate different information and identify stage patients that could be valuable information for clinicians. So based on this idea, we can do more. So we can try, instead of using this idea of monotonic constraints, so just finding progression trajectories that are increasing, we can try to use, so this technically amounts at penalizing the derivative of the progression and just that it's positive everywhere. But we can use more complex constraints to try to simulate or to fit models that can describe dynamical systems that can perhaps illustrate how, for example, protein spreading happens in the brain according to different hypotheses of dynamics of neurodegeneration. So the idea is that we can have a sort of compartmental system. We have different brain areas, and we can make an assumption. So there is a protein that accumulates in the brain that indicate pathologies. And so this accumulation, when it reaches a certain threshold, leads to spreading across the brain connections and so it leads to accumulation also in neighboring regions. So all of this defines some metrics of exchange that can be used to express a dynamical system. So how the speed of change, for example, what we observe in PET imaging data, for example, amyloid deposition, which is a pathological protein in Alzheimer's disease, how it changes over time across brain region according to this dynamical system. And the idea is to learn this dynamical system with the same principle that I described before, just through regression, in this case, through Gaussian processes. So we have developed some theories to fit actually dynamical system through Gaussian processes. This is called gradient matching. And so we applied this kind of approaches to model with the same principle of this time representation that I showed before, to model pathological progression of PET data, amyloid PET data in the brain of patient affected by Alzheimer's disease. So this is the kind of dynamical system where we have some speed, which is parameterized by some matrices where coefficients indicate a speed absolute value, which is modulated by some coefficient that depend over time that indicates actually the shape of the accumulation. So when it starts and when it stops over time. So basically, once we fit this model, we can identify across all the brain region, we can consider across this new time of the disease, we can quantify the strength of propagation from one region to the other. So you see these 3 points are three sample regions, and how they evolve differently, how they interact between each other in explaining how the pathology progresses. And since we have a dynamical system, you can also identify the state space of this dynamical system. So how the brain is going to evolve, the evolution of this uptake, based on the current value, for example, measured at 2 given regions. And since we have a dynamical system, you can integrate it to do prediction. So, for example, over time, here we have a subject, we have 3 time points, we can identify the specific parameter for this subject of pathological accumulation, and then we can predict with an uncertainty. And we see that our prediction are capable of predicting future unobserved testing data for the patients. So ultimately, this system gives us a framework that enables prediction and a way to compare actually biological hypotheses by fitting different kind of dynamical systems, since this is data-driven. And so to integrate different processes, here we are talking about protein accumulation, amyloid, but there is other processes that could be worth investigating for this kind of application. And then how to extend all this theory to simulate intervention scenarios in clinical trials. The idea is that we can still work on dynamical system, but trying to integrate different kind of biomedical information at the same time. For example, different imaging modality, MRI, FDG PET, so metabolism of the brain, amyloid PET, so protein accumulations, and cognitive scores. And the idea is that we can extract some, so using machine learning techniques to identify representations, simplified representations, a sort of Z-scores, and learn how this simplified representation interact over time, for example, by following a given dynamical system. And this given dynamical system in the simplified representation could encode some a priori hypothesis. For example, that this representation that should follow sigmoidal, so monotonic profiles. But this time is not in the data space, as we said before, but this is in this latent representation, so in this simplified space. And the idea is that we have this dynamical system, so we have some sort of system where we project all the multimodal data into this simplified space. Then we impose some dynamical system that describes how this latent representation evolves over time, and we can optimize the parameter of the system to predict the future observation that we have for the patients. So this amounts of being able to integrate dynamical system in the latent space and optimizing this end-to-end, so which means encoding and decoding. So this becomes a variation autoencoder parameterized by ordinary differential equation in the latent space. And we can actually, today, we have the tools, in particular NeuroD, for optimizing this framework end-to-end. And so this becomes a refined way of doing variation autoencoders on time series data. And so this is what we obtain when we apply the model to the data that I showed before. So the model is able, so through by integrating the dynamical system that we learn in the latent space, we can generate trajectories based on the data that describe plausible relationship about how the brain should evolve during the disease. We can see that amyloid starts first to become abnormal, and then it's followed by metabolism and brain atrophy, and also how the clinical score evolves according to the same scale. So we have a complete representation of the evolution of the disease. Also, that can be observed through the different dynamics, so how the different coefficients, how the different representation of the modalities interact together. So this is a bit complex to observe, but we can integrate. So we can observe how they evolve jointly, given a starting point. And what is really relevant is that we can, once we have a dynamical system, we have a full control of the dynamic. And so we can simulate hypotheses. So what happens, for example, if we block completely one of these variables? So we set to 0 the parameters here of the system, for example, for the accumulation of amyloid. So you simulate the fact that we could have a drug that stops completely the accumulation. And we see actually in cascade how the other biomarker evolves to show that we have some trajectories that evolves more like slowly toward pathology. And so once we have this, we can simulate, for example, clinical hypothesis. So what will happen if we have this drug that blocks completely one modality, for example, a pathological accumulation of proteins, and we let the other modality evolve. And so what will be the hypothetical power, statistical power in detect difference between treated and untreated? And so we can simulate different scenario and explain, show that we should intervene at minus 10 years to be able to observe a significant, so a statistical power in the difference for the clinical scores, which is what is used in clinic. And same for if you block 50% instead of total blocking. And so we have different hypotheses, different scenarios. So in this case, we have to intervene a bit earlier. And so we have power curve that can be used to inform actually the setup of new clinical trials in the future. And maybe just a last word. So we tested also this kind of model on clinical data. So this was the model I showed was on research data. We have also validated the model on memory clinic data through collaboration with experts from the Geneva Memory Hospitals, demonstrated we could replicate the same progressions actually on the memory clinic data. And currently we are focusing on models that are able to identify if there are sub-trajectories, so not just assuming that there is a unique trajectory, but multiple one, and decided actually when the multiple trajectory exists. And so applying to Parkinson, we were able to show that in Parkinson data, we can also observe that some score discriminate between two different subpopulations. For example, given the TD score, which is about the tremor symptoms in the population. So this could be insightful to better understand the evolution of neurological disorders. And so to conclude, so this progression modeling is a very powerful discipline that allows to analyze and interpret heterogeneous and high-dimensional data. It could be used to integrate complex hypotheses, and so it can give, like, being used to develop a generative model that can be used to test and simulate novel hypotheses, for example, to help the definition of clinical trial and interventions. And with this, I would like to thank you for your attention. Thank you.